is our posterior confidence measure. This measure essentially says that the more reads you see for an event, the more confident you are about its quantification, and formally it is probability mass around the expected psi region plus minus delta in the psi distribution. And lastly, we have the likelihood function, which is a cross entropy cost function with target variables t. And a good question at this point would be why this particular target function? And the simple answer is that this is the one that worked. So let's see how we perform with this new target function. So here I show a graph where on x-axis I have 5 mouse tissues, and on y-axis I have % variance explained by psi prediction. So the higher the better. So the old BNN model is shown in blue, the old deep neural network is shown in red, and the new network is shown in green. And as you can see that across all the different tissues, we perform much better at estimating psi than the previous approaches. Next, we wish to see if we can integrate CLIP data. CLIP-seq is essentially used to identify binding sites for RNA binding proteins, and we included CLIP data from 15 experiments from different mice tissues for splice factors like RB-FOX, MBNL, SELF, PTB1. And the challenge here was decoupling splicing signal from CLIP data, which is usually noisy and has low coverage. We included CLIP data as features in our model, and each feature was essentially a binary variable indicating presence or absence of a peak in carefully defined genomic regions of interest. So let's see how we perform there. The y-axis is the same as before. The x-axis shows 6 mouse tissues from Mouse Genome Project. And on the left, you see comparison of Bayesian neural network without and with CLIP data. And we see modest improvement when we use CLIP data with Bayesian neural network. And we think the reason for that is that CLIP is essentially providing redundant information, or we are underfitting our BNN network because we wanted to compare effect of a single variable, keeping everything else constant. And on the right, you see the effect of CLIP data when we compare Bayesian neural network versus our new deep neural network. And these networks are actually able to learn psi much better than Bayesian neural network, even when CLIP data is included. Next, we wish to see if we can include splice factor knockdown overexpression data and model splicing prediction for them. So we integrated 7 different splice factor knockdown overexpression data sets, and we encoded their identity as input to the network. And again, with these new kind of data, deep learning models again showed that the data integration and transfer learning actually helps the network to perform much better than Bayesian neural networks, as shown here in this graph. Finally, we wish to see if we can simulate the effect of a splice factor knockdown using these models. And essentially, we do that by zeroing out features related to the splice factor, and we call it in silico splice factor knockdown. On left, you see the correlation plot between, on x-axis, RNA-seq overexpression delta psi value, that's the experimental evidence, and on y-axis, the model predictions. So for the self-1 overexpression data that I'm showing here, which is pretty noisy, we are able to explain delta psi values pretty well. And on right, the y-axis in silico knockdown delta psi values, and you see negative correlation here, which is pretty exciting. And this is the first time this kind of analysis can be done for splicing values, because previous models were modeling delta psi as discrete class variables, making such analysis impossible. Next, we wish to see if we can gain new biological insights using these splicing code models, and this work was done by Matt Gazera in our lab. Here you see on left model prediction of delta psi, and on right the RNA-seq evidence of delta psi in myofibrillar protein NRAP in mouse muscle, and we see reduced exon inclusion upon self-1 overexpression. However, when we did in silico removal of self features, we did not see significant impact on exon inclusion, suggesting an indirect mechanism of regulation. And in line with that, we didn't see any clip peaks upstream of the regulated exon where a self is known to repress exon inclusion. However, when we looked at RBFOX feature removal, we were able to recapitulate the splicing changes, and we know that this exon is indeed regulated by RBFOX2 using RBFOX2 knockdown data. Similar observations were made for MEF2D in mouse heart. And this led to another study where we looked at human T cells and actually found that SELF2 is a potent repressor of RBFOX2, leading to another paper from our lab. It's out in Genome Research right now. So with that, I'd like to conclude. We showed that integrating different experimental evidences like CLIP-seq, RNA-seq, and accurate quantification of splicing signal led to improvement in splicing code models. And we learned that modeling knockout overexpression of splice factors led to some novel discoveries. And for more details, come visit my poster, A289627. Future directions, we would like to scale up using ENCODE and the bunch of RBPs available there. We would like to extend our analysis to human by using GTEx data. Of course, we want to learn more novel biology, disease applications, genetic variations, and we would like to extend our model beyond cassette exons to general LSVs. With that, I would like to thank my PhD advisor, Professor Yusuf Barash, Matt Gazera, who's the co-author in this work, Jody for helpful discussions, and I would like to thank ISCB for providing travel funding so that I could come to this exciting conference. Thank you.
Okay, maybe we have a, yeah, you're ready. Okay.
My name is Yuanhua Huang from University of Edinburgh, and very exciting talk, especially with Dimitri's talk together. And my question is that in Dimitri's talk, he shows that the protein binding is very accurately predict the splicing, and now you showed that sequence predict as well. And so my question is that do you think, have you any ideas about the causality between the protein binding and the sequence and the splicing?
Sure.
Yeah.
Very good question. So we are looking at completely different problems. So Dimitri's problem is actually inclusion versus exclusion. And we are looking at predicting psi values, so it's a regression versus a classification problem. But that aside, what I noticed is that CLIP data, it's not that CLIP data is not informative. If we do, say, a hypergeometric test, we see that different CLIP regions are actually predictive. But we already have a bunch of sequence-based features, so the information that CLIP is providing, we think is redundant to that. So it's informative, but that does not necessarily translate to prediction power.
Mm-hmm. Thank you. Can I ask another one? I read the paper that you used the deep learning to predict the splicing, but I got a feeling that deep learning's benefit is that it can define the features itself. So like you give a window of the sequence and then it goes to predict it automatically. And here in your model, it's like you define 1,000 features and then do the prediction. And actually, even with the simple linear regression, it can predict very well. Although yours is better.
My life would be so much easier if we could predict splicing from sequences. But the reality is that we are working on convolutional neural networks that would try to use sequences more directly. But right now, with the current state of things, it's not, at least for our problem, it's not possible to train directly from sequences to predict splicing.
Okay, thank you.
Okay, thank you very much. So let's wrap up. So first of all, let's thank you all the speakers from today. So a big round of